Lovely jubbly, this is question 6 out of the class questions book, and we're on human reproduction, which is one of my favourite topics, and it's about sperm, which is even better. So there we go. The micrograph below shows a human sperm cell, and we've got a scale bar, which immediately makes me think, oh, calculating. So calculate the actual length of the sperm cell. So you can do it in 2 stages and use the scale bar to work out the magnification, and then do the actual is image over magnification. But that's a bit of a lengthy way of doing it. You're very welcome. If that's the way you understand it, that's the way you should do it. But what we want to know is how many times that number goes into that number. So how many lots of that scale bar could we have? So I'm going to measure my sperm cell, and it is 100 millimetres long, because we always measure in millimetres. So 100 millimetres, so that's that distance. And then I need to measure my scale bar to know how many times it will go into it. That's a division sum. So yeah, that's 19. So 100 over 19, and then each one of those, that tells me how many times that scale bar fits in, and each time it fits in, it's worth 5 micrometers. There's no conversions to do here, which is lovely, whereas if you do it the other way, of course you have to. So 100 shared by 19 means that scale bar fits in 5.263 blah 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 times, and I'm going to multiply that by 5. And I'm going to do that to I'm going to do it to 2 decimal places, because that's what I always do. So I've now got 26.3. My next number after the second decimal place is 5. 26.32 micrometers. That was a nice easy starter. So, mitochondria in the sperm do not enter the secondary oocyte at fertilisation. So, sperm mitochondria don't get into the oocyte. What does this suggest about the origin of all mitochondria in an organism? Well, if they're not from the sperm, they've got to be from the oocyte. So, all mitochondria are from the secondary oocyte of the female. So again, this is an example of scaffolding in a question. So we've established that the mitochondria are all from the female, and a woman has inherited a mutation in her mitochondrial DNA, resulting in mitochondrial disease. Her husband and father do not have the disease. Complete the table to indicate which of her relatives will suffer from the same mitochondrial disease. So, we've said that all the mitochondria are from the female, so her son is going to inherit his mitochondria from his mum. Her daughter is going to inherit the mitochondria from the mum. Easy peasy. Daughter's children, so the daughter's got all her mitochondria, and she's female, so she's going to pass them on to her children. But the son is only going to contribute a sperm to his children, no mitochondria then. So they won't suffer from mitochondrial disease unless he's married somebody with it, obviously. So, the inheritance of mitochondrial diseases can be prevented using the technique outlined in the following diagram. So, this was something that was in the news, the successful 3-parent babies. So, we've got a donor egg with healthy mitochondria. Take the DNA out. But parent egg with abnormal, patient egg with abnormal, we remove her DNA from the secondary oocyte and put it into the donor. So that's her nucleus into a different cell with healthy mitochondria. And then we're fertilising it with a partner sperm and implanted back into the patient. So not the donor, the patient. We're not talking surrogacy here. Use all of the information provided. Here we go again. Explain why a child produced by this method is a 3-parent baby. So we just need to say who the parents are, really, I think. So, the nuclear DNA from the patient is one parent. This is the genetic mother. We've got the nuclear DNA from the sperm. So this is going to be the genetic father. And we've got donor cell with healthy mitochondria from a different female. So that's the third. So we've got 1 parent, 2 parents, 3 parents. Super duper. Okay. Oh, calculations. Love a bit of calculating. Okay, Hemocytometer can be used to calculate the number of sperm in a sample of semen. I don't think this is a job for you lot because you don't like using microscopes. So, Hemocytometer is a microscope slide with a rectangular chamber engraved with a grid. The cover slip is supported over the chamber, so we've got that down there. And the depth of the chamber is known. The number of sperm cells in a specific volume of semen can be counted. So we've got a grid with a known area, and we've got a depth as well, so we've got volume. So the counting area is observed using a microscope and the number of sperm cells is counted. The sperm are killed before counting. So why can we not count live sperm? What do we know about live sperm? They can swim. So if they're all wriggling around on here, then you're not going to be able to count them because you can't mark them to say we've counted that one already. So live sperm would move from one square to another. Okay,